welcome to the 11th talk of the webinar series on genetics and evolution, organized by the Journal of Genetics. I'm very happy to welcome Professor Damian Dowling. He works at the School of Biological Sciences, Monash University. Professor Dowling got his PhD from the University of Melbourne, working on the reproductive behavior of bird systems. He then went on to do a postdoc at the Uppsala University, Sweden, where he worked on Drosophila system, working at the nuclear mitochondrial coevolution and its effects on various life history traits. He then went on to do another postdoc at the University of Western Australia, where he worked on the CLIP system. And since 2009, he has been at Monash University. Professor Dowling has made fundamental contributions to our understanding of nuclear cytoplasmic interactions. He has studied in great detail the intergenomic collaboration and conflicts between the nuclear and mitochondrial genes and how these affect aging, sex differences, and a host of other interesting questions. Today, he's going to be talking about mitochondria, life histories, and the evolution of sex differences. Professor Dowling.
Many thanks for the wonderful introduction, Prasad, and also for the invitation to speak today. So I hope you can hear me loud and clear. I thank Prasad, the Journal of Genetics, and the Indian Academy of Sciences, and all the organisers of this webinar series for this invitation and opportunity today. The research I'm going to present is the culmination of several years of studies that have been conducted by students and postdoctoral researchers in my laboratory. And quite a bit of it has been done with international collaborators as well. And here I just want to start out by acknowledging these researchers and their hard work that's generated the insights I'm going to present today. Many of the studies actually come from PhD students that have studied in my group over the past few years. I'll be highlighting research today by Florencia Camus, who's now at University College London, Venkatesh Nagarajan Rada, who's now at the University of Sydney, as well as Rebecca Vaught, and who's back in Alabama, USA, and Winston Yee, who is my laboratory manager. Yonsei Wolf was also an important contributor to much of the research I will present today, and he was a postdoc in my lab for several years. We've also collaborated with researchers in Europe and New Zealand and the UK. And I'm going to be presenting some of this research today, in particular research I've done with Neil Gemmell and Dan Tompkins, as well as David Clancy, Ted Morrow, and Paolo Innocenti. So, unless you're a microbiologist, there's a good chance that when you're pondering the evolution of life, you're thinking about lineages that lie on these branches of the phylogenetic tree. So these are our eukaryotes. They're structurally more complex than the prokaryote domains. Their DNA is enclosed in a cell nucleus. They have a cytoskeleton and membrane-bound organelles, which include the mitochondria. In green lineages, they have the chloroplast. Now, a key point here is that one of the key defining features of the eukaryotes are mitochondria. Indeed, the last eukaryote common ancestor had mitochondria. And so this acquisition of mitochondria is very likely to have been a key factor in mediating the evolutionary trajectories of complex multicellular eukaryote life. These mitochondria presumably enabled that eukaryote to harness an extremely efficient means of energy conversion. So this is achieved by oxidative phosphorylation, which takes place within the mitochondria of the cell. So these mitochondria are loaded into the cell's cytoplasm. And the cool thing about this process is that it consists of a series of enzyme complexes, here denoted on the right hand of this panel. And these enzyme complexes consist of subunits that are encoded by not one, but two distinct genomes. Now, this is because the mitochondria have retained their own genome, which is highly streamlined in animals. It consists of just 37 genes, 13 of which encode subunits of OXPHOS. So here you see these subunits marked out by red circles in 4 of the 5 enzyme complexes. These are our mitochondrial encoded subunits. And they must interact with subunits that have been encoded by the nuclear genome. They're the subunits, the circles that are marked out in blue. And because the mtDNA genes serve such vital functions, and therefore should be under intense purifying selection, for many decades, evolutionary biologists assumed that the genetic variation observed within the mitochondrial genomes, both within and between populations, they assumed that this genetic variation had accumulated under a neutral equilibrium model and was not important in shaping the dynamics of adaptive evolution. As such, mitochondrial genomes were harnessed as quintessential markers for making evolutionary and phylogenetic inferences, all under the assumption that that variation was neutral. And the role of the mitochondria as mediators of evolutionary processes was generally ignored. So recent years have seen this paradigm largely overturned, and the emergence of a new and active discipline of study has come to be termed mitonuclear ecology. Experimental evidence has now clearly shown that mitochondrial genomes often harbour genetic polymorphisms that change the phenotype. We'll call this functional genetic variation. And here's just one example from my own work. Now, this study is 11 years old now. Myself and colleagues sampled 25 females from a laboratory population of fruit flies, Drosophila melanogaster. These females became the founders of their own mitochondrial strains. Let's call them mitochondrial Eves, if you will, in which we placed the cytoplasms of each female alongside a standardized nuclear genomic background, just using standard techniques of backcrossing. We then measured both the reproductive success and the lifespan of females of each of these mitochondrial strains. We also sequenced fragments of 3 mitochondrial genes to see whether flies of each strain actually carried different mitochondrial haplotypes. Now, a sequencing effort which we carried out on about 15% of the mitochondrial genome, it revealed a solitary SNP at position, I think it was 204 of cytochrome B gene. And this SNP resulted in an amino acid transition from cysteine to tyrosine. Furthermore, what became clear is that females of the strains that carried the tyrosine variant, they had both higher longevity and higher reproductive fitness than females of the strains that carried the other cysteine variant. Now, I found this result striking because our original sampling of the ISA females that contributed to creating these lines suggested that the tyrosine variant was actually at much lower frequency within the population. But only 8% of the flies that we initially sampled carried the tyrosine variant. So most of the flies in the population seem to be carrying this worst performing variant, the cysteine variant. And that's not what you'd expect under a simple argument of Darwinian selection favouring the genotypes that it existed. So this result really provided the baseline question and motivation for much of the research that we've conducted in my research group over the past decade. And that question is, how has this genetic variation, this functional genetic variation, been able to accumulate? Evolutionary theory suggests a possible role for both adaptive and non-adaptive processes in driving the accumulation of this variation. For instance, the variation could represent a non-adaptive pool of deleterious mutations, and indeed it was long hypothesized that the high mutation rate and lack of recombination in the maternally inherited mitogenome provided the conditions that would be ideal for the accumulation of deleterious mutations under a process akin to Muller's ratchet. Now, the alternative is that at least some of the functional variation is adaptive and retained in the genome because it benefits the beholder. Indeed, a series of studies emerged over the past 15 years that started to report correlations between the mutational patterns found in the mitochondrial genome and global climatic zones. These patterns, while intriguing, were based on correlations only, but they nonetheless suggested that the genetic variation found within the mitogenome might be involved in the dynamics of climatic evolution. But little experimental evidence exists. So my lab has set out to try to answer this question. What genetic processes lead to the accumulation of functional genetic variation within the mitochondrial genome? And we try to answer this question by injecting a fresh approach, an experimental approach grounded in evolutionary ecology. So we deploy experimental designs in which we can really start to drill down and partition out relative contributions of adaptation and mutation accumulation in contributing to the functional variation that we see within the mitochondrial genome. And today I'm going to take you through a few case studies from my lab in which we've harnessed natural levels of mitochondrial variation that exist within and across natural populations of the fruit fly Drosophila melanogaster. And we've used experimental approaches to try to investigate roles of the non-adaptive and adaptive processes in shaping the mitochondrial genome. I'm going to start by presenting a study in which we explored the question of whether the standing variation found within the mitochondrial genome of Australian fruit flies might be shaped by climatic selection. And the ecological framework that we harnessed is this latitudinal climb of fruit flies that exists along the east coast of Australia. Now, we know from prior work that strong climbs exist for body size, heat, and cold tolerance along the eastern seaboard of Australian flies. And there are molecular underpinnings of these phenotypes found in the nuclear genome that match these phenotypic climbs. So as you move south along the east coast of Australia, the flies become larger, less heat tolerant, but more tolerant of cold stress. So we harness this climb to explore the adaptive significance of mitochondrial genetic variation found within Australian fruit flies. And essentially what we were testing was a new hypothesis that we call the mitochondrial climatic adaptation hypothesis. We formulated two clear predictions that we aim to test. The first prediction is that the genetic variants that we found in the mitochondrial DNA sequence within the Australian metapopulation of flies would exhibit linear associations with latitude, such that some haplotypes might predominate in cooler temperate areas of Australia, down here in the south of Australia, and others at low latitude subtropical northern populations. So frequencies of haplotypes should exhibit linear associations with latitude. That was our first prediction. The second prediction is that thermal tolerance phenotypes should map to the mitochondrial genotype, such that haplotypes that might predominate in the subtropical north of Australia should confer greater resistance to heat stress, while those that predominate down in the southern temperate parts of Australia should confer increased tolerance to cold resistance, to cold stress. I'm going to run you through the results of a paper where we tested these predictions, and the paper was led by PhD student who's now an alumni from my lab, Forensic and postdoc Yonshi Wolf, in collaboration with here at Monash. And the first thing we need to do in order to test these predictions is to gauge levels of mitochondrial DNA sequence variation across populations spanning the east coast of Australia. So we collected flies from these 11 different populations that you see starred here on this map. And we then examined full mitochondrial sequences across pooled samples of basically 90 flies from each of these populations. So we used the pool sequencing approach, and using that approach, we identified 15 SNPs in the mitogenome that appeared to delineate at least the northern and the southern populations. So using these 15 SNPs, we could then probe for haplotype diversity and actually start to estimate the frequencies of the haplotypes within the populations by designing a custom genotyping array based on the SNPs. And we used this assay to genotype all the females that were collected from the field. And what we observed was that there's essentially two groups of haplotypes here, an A group in red and a blue group in blue. And each haplogroup was delineated by a major haplotype, so A1 and B1. And these were indeed differentiated by 15 SNPs. They were all synonymous SNPs. These SNPs don't change the amino acid sequence. So now we had the power to genotype the flies. We could start to test our predictions. Let's have a look at prediction one. First, that the population frequencies of these haplotypes would exhibit latitudinal climbs. And indeed, we found strong evidence for this prediction. Here we can see nice linear correlations between latitude on our x-axis and our haplotype proportions on our y-axis, both on the left for haplotype A1, and then just the inverse correlation that you see on the right for B1. So the A1 and the B1 haplotypes is present in every population that we sample, just at a different frequency. And so you see that the A1 haplotype predominates in the low latitude subtropics, whereas the A1 haplotype predominates down in the temperate areas of Australia, such as Melbourne, where I live. So now we've confirmed prediction one, we could move on to testing prediction two. This prediction is that the thermal tolerance phenotypes would map to each of these haplotypes, such that the northern predominant A1 haplotype would be associated with increased tolerance to heat stress, while the southern predominant haplotype, the B1 haplotype, would be associated with increased tolerance to cold stress. So in order to test this prediction, we decided to create genetic strains of flies that differed only in their mitochondrial haplotype. We picked 8 females, 4 from a city in Australia called Brisbane, and the other 4 from Melbourne. Now, these cities are separated by thousands of kilometres. Within each of the city, we made sure that 2 of the females carried the A1 haplotype and 2 carried the B1 haplotype. And then, taking advantage of the maternal inheritance of the mitochondrial genome, we simply backcrossed each of those haplotypes of each of these females into an isogenic nuclear background. So this is a genetic background that's basically devoid of any genetic variation. It's clonal or inbred. And we did these backcrosses over a sequential period of 24 generations, creating these 8 different strains, 4 of which carried the A1 haplotype, 4 of which carried the B1. Now, at this point, we decided to resequence the strains just to make sure that there had been no contamination events and to ensure that the 4 A1 strains indeed carried the A1 haplotype and that the 4 B1 strains carried the B1 haplotype. And the resequencing effort indeed revealed that each of the 4 A1 strains carried the unambiguous A1 haplotype, but there was a little bit of a twist with respect to the B1 strains. The B1 strains each carried these 15 SNPs that distinguish B1 from A1. But each of our B1 strains carried an extra 1 to 4 SNPs. So that in effect we had sampled 4 B1 strains that each carried a unique subhaplotype of the B1 haplotype. We called these B1A through to B1D. Some of the SNPs that distinguish the B1s changed the amino acid sequence as well. But they all were separated from A1 by these 15 common SNPs. So once we had the 8 strains, we submitted the male and female flies of each to 2 thermal challenges. Firstly, exposure to an extreme heat stress, and secondly, exposure to an extreme cold stress. So in the extreme heat stress challenge, we essentially placed flies into small microtubes and put them into a water bath, which was heated to 38.5 degrees Celsius. And we recorded how long it took until those flies fell into a coma. In the cold stress assays, we placed flies in a cold-induced coma by exposing them to 0-degree temperatures for 4 hours, and then we recorded how long it took for those flies to wake up from that cold-induced coma. Let's have a look at the results. So we'll start by having a look at the capacity of the flies of each haplotype to withstand extreme heat stress. And what you can see here is that flies that carry the A1 haplotypes, so here in red, had a superior response, taking longer to fall into coma than any of the flies of the B1 subhaplotypes in blue. This might become clearer if I add a horizontal red bar across the box plot here, so that you can see that none of the means and standard errors overlap for those 2 groups. Remember that this A1 haplotype, denoted in red, is found at higher frequencies at our low latitude subtropical populations. That is, it's present in the field, we find it in the warmer subtropics. And it also is associated with higher tolerance to extreme heat stress in the laboratory. We also had a look at the performance, the heat tolerance performance in each of the sexes, in males and in females, and you can see how the performance differs for each of these haplotypes on the right-hand side of this screen. And you can see that generally performance in males mimicked performance in females despite strong sexual dimorphism. So males are generally more heat tolerant than are females, but the haplotype A1, for instance, which was superior in females, was also superior in males. And we didn't get much crossing over of those reaction norms across sex, with the exception of one haplotype, the B1D subhaplotype. Let's have a look at this one here. Here you can see that generally males, if males carry the B1D subhaplotype, they basically have a female-like heat tolerance phenotype. And this was particular, it was exclusive to males carrying this subhaplotype. Males are basically duds if they have this subhaplotype, or when it comes to heat tolerance or resilience to extreme heat stress. Now that's quite fascinating, particularly in light of the concepts that I'll talk about in the second half of this talk. Let's now have a look at how these haplotypes responded to extreme cold stress. Here you can see the reverse situation. The B1 haplotypes are now outperforming the A1 haplotypes and associated with more rapid recovery following a cold-induced coma. And indeed, it's the B1 haplotypes that are found at higher frequency in high latitude, colder populations of Australia. So, in sum, we confirmed each of the two predictions of the mitochondrial climatic adaptation hypothesis for this metapopulation of Australian flies, providing some of the first definitive experimental support for the idea that thermotolerance responses of each haplotype, when measured using very carefully controlled experiments in the laboratory, map closely to the spatial distribution of these very same haplotypes in the wild. So that provides pretty strong evidence that climatic selection can shape the standing genetic variation found within the mitochondrial genome within a population, thus implicating the mtDNA as an important player when it comes to evolutionary responses to climate change. So that wraps up the part of the talk where I address the case for adaptive selection in shaping sequence variation we found in the mitochondrial genome. In the second part of the talk, I want to discuss the potential for deleterious mutations to accumulate within the mitochondria. And I want to focus on a special subset of mutations that are specifically male harming. So natural selection is like a sieve. The sieve sits at this boundary between two generations, and it basically has the job of screening the good mutations from the bad, only allowing those mutations that are benign in effect or positive in effect to pass through the sieve and fall into the gene pool of the next generation. Now this sieve works on all of our DNA, both nuclear genomic, but also our mitochondrial DNA. But when it comes to the mtDNA, there's a catch. And that catch is related to the strict maternal inheritance of mitochondria. In 1996, Frankenhurst proposed a simple genetic model, and it's based on the maternal inheritance of the mitochondria. Now, they realized that because of maternal inheritance, all of the quality control on the mtDNA sequence has to happen through females. Therefore, the evolutionary fate of any mutation in the mtDNA sequence is determined solely by fitness effects in females. That is, if a mutation in the mtDNA was to arise that did not exert any effect whatsoever on female function, it would be free to fall through the selective sieve, even if this same mutation had very negative effects on males. And over evolutionary time, this should, in theory, lead to the build-up of a pool of mutations that negatively affect male function, but with no effect on females. That is, it should lead to a male-specific mutation load in the mitochondria. Neil Gabel, Victoria Metcalf, and Fred Allendorf called this the mother's curse. Now, the mother's curse hypothesis predicts that we'll be able to identify mutations in the mtDNA sequence that specifically cause harm to males and males only. The question is, have we identified any such mutations? Well, we know that there are plenty of mutations that fit that billing. We call these cytoplasmic male sterility mutations, and they map to the mtDNA sequence. Frankenhurst envisaged that when it came to animals, that male sperm quality and fertility would be traits that might be most susceptible to the effects of mother's curse, and indeed, several examples have now arisen of mitochondrial mutations with fairly catastrophic effects that are limited to the male fertility phenotype. So examples have come up from both lab studies of mtDNA variants in flies and in mice, but studies have also revealed similar mother's curse mutations affecting fertility in wild populations of European hares, and also evidence of mother's curse mutations in humans. Now, these mutations are not specific to any one region of the mtDNA sequence, but they appear to be dispersed across several genes. What I find really remarkable about all of these cases is that for the most part, the researchers that identified them really stumbled upon them by chance. They were serendipitous findings. Searching for these mother's curse effects was not the goal of the individual studies that reported the effects. And basically they found these effects because the effects of the mutations were so great, essentially knocking out function, causing complete male sterility in many cases, for instance. But what we currently lack is a more nuanced and broader understanding of whether this evolutionary process of mother's curse has more pervasive effects on the life histories of animals, shaping performance and function of males more generally than just these few isolated examples of loss of function mutants that severely impact male performance. Now, solving this question requires a general and systematic screen of mother's curse effects across populations. And so, how might one conduct such a screen? Let's walk through a predictive framework for how we might test the mother's curse hypothesis. What would we expect if we went out into the wild and sampled a set of mitochondrial haplotypes, here depicted by circles, from different localities within the distribution of a given species? So under the mother's curse hypothesis, we predict that purifying selection will be very effective at eliminating harmful mitochondrial mutations from accumulating when these mutations are affecting females. But because of maternal inheritance, purifying selection will miss the class of mother's curse mutations that are explicitly male harming in their effects. And these mother's curse mutations, depicted here in yellow lightning bolts, should therefore be free to accumulate. And they should accumulate at different rates within each of these different populations, because each of these populations are evolving along their own specific trajectories. Therefore, the haplotype should be accumulating different numbers and identities of these mother's curse mutations. And therefore, each of the haplotypes will be associated with a different mother's curse mutation load, which is associated with different severity in the effects on the male phenotypes as we go from one to another. Okay. So the haplotypes will differ in their number of mother's curse mutations, the sites of mutation, the particular polymorphisms involved, and their associated severity of effect. So, in theory, to uncover these mutation loads, all we really need to do is to sample the haplotypes and measure their associated effects in each of the sectors. So this all boils down to one simple but key prediction of mother's curse, and that is that the genetic variation that accumulates across mitochondrial haplotypes will have larger effects on phenotypic expression in males than in females. That is, male phenotypes should be more sensitive to mitochondrial variation than will female phenotypes. So over the past decade, in my lab, we have been trying to test this prediction by assessing the phenotypic performance of naturally sourced mitochondrial sampled from wild populations of fruit flies. And we've principally used a panel of strains in which we've sampled 13 different mitochondrial haplotypes sourced from basically all the different corners of the globe. And we've placed these 13 different haplotypes alongside a standard nuclear genetic background that lacks genetic variation, such that the only genetic variation found across these 13 genetic strains is found within the mitochondrial DNA sequence. This gives us a powerful way to disentangle effects of mitochondrial genotype from effects of the nuclear genotype. Furthermore, each of the haplotypes is actually independently replicated across that panel, providing us with a really powerful way to partition effects unambiguously to the mitochondrial genome, separating out other potentially confounding sources of variation. So these haplotypes differ in their level of molecular divergence. Some are separated by only a very small number of SNPs, others are delineated by up to 40 SNPs with other haplotypes in the panel. And using this panel of haplotypes, we've measured a number of phenotypes across 3 levels of biological organisation. First, molecular phenotypes, then physiological phenotypes, and also life history phenotypes. So when it comes to molecular phenotypes, we've primarily measured effects of the different mitochondrial haplotypes on patterns of gene expression across the whole nuclear transcriptome, as well as across single key mtDNA protein coding genes. When it comes to physiological phenotypes, we've measured both the metabolic rate of flies of each sex, as gauged by levels of carbon dioxide respiration, but we've also had a look at the underpinning mitochondrial bioenergetic traits, looking at some fine-scale respirometry. When it comes to life history phenotypes, we've measured traits like longevity and reproductive success. And I'll be quickly running through some of our key findings across these studies, and in each case we calibrate the results against the central prediction. Mitochondrial haplotypes or haplotype-mediated effects should be larger in males than they are in females. The first thing we ever did with the panel was examine how different mitochondrial haplotypes may affect regulation of patterns in nuclear expression. And this study is now going back quite a few years. It's published in 2011. And it was led by Paolo Innocenti in collaboration with Ted Murray. And because of its age, we used a microRNA approach rather than RNA sequencing. So a microarray approach rather than RNA sequencing to profile the nuclear transcriptomes of both males and female flies that differed only in their mitochondrial haplotype. And what we found was surprising, to say the least. We found that the expression of around 1 in every 10 nuclear genes of a male fruit fly was sensitive to the identity of the mitochondrial with which those nuclear genes sat alongside. That's to say, cryptic variation harbored within the mitochondrial genome affected the regulation of over 1,000 nuclear genes in males. In females, the situation was completely different, with only 7 genes in total exhibiting signatures of differential expression. 7 nuclear genes when placed alongside the different. Furthermore, of the nuclear genes in males whose expression was sensitive to the mitochondrial haplotype, more than 1/3 of these exhibited expression patterns that are localized to the male reproductive traits, that is, testes, the accessory glands, and the ejaculatory duct. It seems to be that the genes that are involved in encoding male reproductive outcomes that appear to be some of the most sensitive genes to sequence variation found in the nuclear genome. So these results provide strong support for our key prediction that haplotype-mediated effects would be larger in males than they are in females. Moving on to physiological traits, and here I'll focus only on the results of metabolic rate. So this is work led by Venkatesh Nagarajan Radha, who completed his PhD in my group last year, in 2019. And we found that male biases in mitochondrial haplotypic effects on metabolic rate were quite large. So here on the left-hand side of the panel, we have the haplotype-mediated effects on metabolic rates in males and in females, here to the right in red. In short, and you can see this clearly by looking at the interaction plot, each of these lines represents a different mitochondrial haplotype. We have the effects in males and the effects in females. You see that these reaction norms fan out in males. It shows that there is much more variation across the haplotypes in their effects on metabolic rate in males than there is in females. I think the variation was something like 2-fold higher in males than in females. And again, that evidence is completely consistent with this key prediction of mother's curse. Let's look at what's happening at the life history level. Here, this is work led by Ferencia Camus in collaboration with David Clancy. Mitochondrial genetic effects on longevity were also highly male biased. Here again, we have the effects on males on the left-hand side of the panel and the effects on females on the right-hand side of the panel. And you can see that there's a lot of variation across our haplotypes in male longevity. In female longevity, there's a little bit of variation, but many of our haplotypes seem to converge on the same trait value, basically get to a flat line for many of these haplotypes. So you have a statistically significant increase in levels of mtDNA effects on male longevity. What does this mean? Let's step back and consider what it actually means. It means that there's presumably genetic variation in the mitochondrial genome that has little effects whatsoever on the longevity of females, but which exert quite profound effects on male longevity. So the mitogenome harbors mutations that are affecting only longevity outcomes in males. And this variation, which presumably reflects the existence of mother's curse mutations, could help explain the sex gaps we see in lifespan across animals and differences in life expectancies we see in humans as well. We also found some fascinating genetic correlations across our mitochondrial haplotypes, which again, these correlations were limited to males, implying that the mutational variation driving these correlations consisted of male harming variants that had accumulated under mother's curse. So here you can see, and this is again work led by Venkatesh, that the haplotypes that are associated with high metabolic rate, sorry, my mouse is the haplotypes that are associated with high metabolic rate in males are the haplotypes that are associated with the shortest lifespan. It begs the question, is this a signature of a live fast, die young trade-off that's regulated by the mitochondrial genome and entwined in the mother's curse process? It's a fascinating question. This sets the seed for addressing that in future studies. So far, all of the studies that I've uncovered have supported the key prediction of mother's curse, that is, male bias in haplotype variation. As such, we figured we would test one last set of traits, and that is reproductive success in each sex, confirm the male bias, and then we have definitive validation of our hypothesis. Yet when we sampled the effects of the mtDNA haplotypes in our panel on reproductive traits that we had measured in each of the sexes, the results were not clear-cut at all. They were definitely not as predicted. We didn't find the predicted signature of a male bias in effects. This was the same regardless of how we measured reproductive success, be it via egg number, adult offspring production, or the proportion of eggs that converted through to adults in females, or be it via assays of reproductive success that were confined to a really short period of mating early in life at the reproductive peak, or whether we gauged reproductive success over a much longer period of time that stretched across, say, for instance, the first 10 to 12 days of life. Regardless of how we measured reproductive success, we did not find that predicted male bias. Now, this proved very confusing to us, but we gained some key insights into what might be happening when we began screening for the possible existence of genetic correlations across the mitochondrial haplotypes for these different components of reproduction. And here's what we found. This work, again, was led by Forensic Kamus while she was a PhD student in my lab. For the different reproductive traits that we measured within a sex, what we found is that the mitochondrial haplotypes that conferred the highest trait value for one trait, these haplotypes generally conferred the highest values of the other traits, such that the direction of intrasexual genetic correlations across mitochondrial haplotypes was positive. Now, in contrast, when looking at the mitochondrial genetic effects on the reproductive traits across different sexes, what we found was that those haplotypes that conferred the highest reproductive outcomes in females were generally the haplotypes that conferred the poorest outcomes in males. That is, the direction of the intersexual genetic correlations across mitochondrial haplotypes was negative. So when it comes to reproductive outcomes, the haplotypes are in effect appearing to act sexually antagonistically. They seem to harbour mutations that depress male reproductive outcomes, but these same mutations, they're not benign in effect in females, as predicted by the mother's curse hypothesis. These male-harming mutations appear to directly benefit female reproductive outcomes. Now, this result was surprising, and thus we went back to some of our other traits to see if these same intersexual negative correlations were evident in those traits. And indeed, Venkatesh detected a clear antagonism that permeated to the level of metabolic rate. Here we see that haplotypes that are associated with high metabolic rate in females are associated with the lowest metabolic rate in males and vice versa. So a signature of sexual antagonism for the metabolic rate as well. Now, this leads to a reformulation in the way that we think about mother's curse mutation. Let's return to the depiction of the selective sieve that represents the action of natural selection in screening mutations that enter the gene. Now, when applied to the mtDNA, the sieve sorts the good out from the bad, and it only allows the good to fall through, but it misses this class of mutations that we called mother's curse mutations that were male-harming but benign in females. Well, our study suggests there's an extra class of mother's curse mutations, those that are bad for males but explicitly beneficial for females. Here they're depicted in yellow. Now, due to maternal inheritance of the mtDNA, these mutations should be under positive Darwinian selection and should thus quickly accumulate within populations. If I have time, and I think I have about 5 more minutes, I just want to briefly touch on a case study of one particular haplotype that comes from Brownsville, Texas, and we call it the Brownsville Hypotype. Now, this haplotype harbours a SNP in the cytochrome B gene which results in an amino acid transition, and it's associated with male sterility. In the nuclear background that we keep these flies, in fact, Brownsville, this haplotype, is completely male sterile. Incidentally, no matter what nuclear genetic context or environmental context we measure this Brownsville haplotype in, it's always associated with male infertility or male subfertility when it comes to components of reproductive success. But it's a very decent performer in females. Here we have our reaction norms of 13 different haplotypes from our panel for reproductive success of 3 days early in life, day 1, day 4, day 8 in males and females. The performance of the Brownsville haplotype is depicted by the thick red line. It's completely sterile in males, but in females it does all right. In particular, early in life, it's quite a high performer. And at day 8, it's also one of the higher relative performers. And here you can see that no matter the context in which we measure this Brownsville mutation, it always does poorly against our other subhaplotypes when it comes to male fertility phenotypes. Intriguingly, however, males that harbour this male fertility impairing haplotype actually enjoy longer lifespan than males that harbour any of the other haplotypes. And while the Brownsville haplotype is associated with high reproductive success in females, it incurs a longevity cost to females. So females that harbour Brownsville suffer shorter lifespans than females that harbour any of the others. We have some complex patterns of antagonistic pleiotropy and negative pleiotropy across the sexes. Again, here what we can see is that Brownsville, despite being male sterilising, is actually associated with the highest juvenile viability of all our haplotypes that we've measured in our panel, both in terms of rate of eclosion from pupal cases and in terms of egg to adult viability. So here we have on the left data collected by Yonchi Wolf, showing that Brownsville outperforms 2 other haplotypes when it comes to pupal viability. And here's data from Florencia Camus showing the performance of Brownsville is superior to the performance of all other haplotypes when it comes to egg to adult viability. So, this is a fascinating question. Could the male sterilising Brownsville haplotype actually be under positive Darwinian selection in populations because it benefits females through increased reproductive success and juveniles through heightened juvenile viability? Now, we used an experimental evolution approach, which I can very quickly take you through, and will be the last couple of slides of the talk. Specifically, we wanted to test this question. So this is work led by former lab postdoc Yonchi Wolf in collaboration with Neil Gamble and Dan Tompkins in New Zealand. Now, to initiate the experiment, we commenced with 6 large replica populations of a large population of fruit flies originally collected in Benin, Africa. It's known as the Dahomey population. Now, we introgressed the Brownsville haplotype into 3 replicate populations of Dahomey, while the other 3 replicate populations carried the native Dahomey haplotype. So in effect, we had 6 replicate populations. Each had high levels of standard nuclear variation, representative of what is found in the ancestral Dahomey population. Now, the only difference across populations is that half of them carried the Brownsville haplotype, the other half carried the Dahomey. We could then use these replicate populations to create new experimental populations, seeded with varying starting frequencies of the Brownsville haplotype. So we initiated populations in which the Brownsville haplotype started at either 0% frequency, so it wasn't in those populations, 25% frequency, 50% frequency, or 75% frequency. And then over the ensuing 10 generations, we simply tracked the viability of each of these populations, and we tracked changes in the frequency of the Brownsville haplotype relative to its dark competitor. Here are the results, and I'll walk you through them now. And what we found was very interesting. When seeded into populations at greater than 50% frequency, here depicted by the blue and orange data points, the Brownsville haplotype led to sustained reductions in population viability over sequential generations. These reductions in viability were clear by generation 5. And what's remarkable is that despite there being a well-segregated nuclear variation in these experimental populations, we observed no compensatory response of the nuclear genome across the 10 generations of the experiment. There was no restoration fitness. The depressed fitness, and it was about 10% fitness depression, held across the experiment. No compensatory response. Now, what was perhaps most remarkable about this experiment is that the negative effects of the Brownsville mutation on population size went hand in hand with sustained increases in the frequency of the Brownsville haplotype across the experiment, at least in the populations that were seeded by greater than 50% Brownsville haplotype. So this is seen in the bottom panel. Now, what this indicates is that the Brownsville haplotypes are able to drive into a population, presumably under positive selection in females and juveniles, despite their really negative effects on male fertility that actually lead to suppressive effects on population size. So despite the suppressive effects on population size, this Brownsville haplotype is presumably under positive selection. Well, I think it's time for me to wrap up and summarise. And so I'll end my talk with a brief summary of 3 key points. Firstly, it's now become clear that genetic variation exists in the mitochondrial genome that modifies the phenotype. Secondly, the experiments I've presented today indicate that some of this genetic variation is likely to be adaptive, some male maladaptive, and some of it's even likely to be sexually antagonistic. Finally, the results provide new insights into the role that mitochondria likely play as contributors to evolutionary adaptation and to trajectories of human health, including the genetics of male infertility and female biases in longevity. So I'll wrap up there and thank you all. Thanks to the wonderful members of my lab, both past and present, who have been responsible for virtually all of the research that I show you today. And again, sincere thanks to Prasad for the invitation to present it as part of this seminar series. Thank you.
Thank you, Damian. Thank you so much. I'll start with an interesting comment that has come in. The Brownsville study is fascinating and adds a whole new dimension to the title of Bob Dylan's song, Brownsville Girl. So, following up on that, the Brownsville system, this is from Amitabh Joshi, the Brownsville system might be an interesting system within which to quantify individual versus group selection, since haplotype frequency within a population increases while population viability decreases.
That's a really good comment. That's a question that's never been asked before when I've given a seminar, so I'm a bit stumped, but I can see exactly what you're talking about, that it might offer a perfect tool to measure levels of selection and group selection in an experimental evolution context. Yes, great insight.
Again, a question from Amitabh. For testing the mitochondrial haplotype effects on thermal tolerance, would it not be better to try it out in at least a small set of different genetic backgrounds? There could be haplotype by genome interactions.
Yeah, this is also a wonderful question. We went for the easier proof of concept to start with. And then when we confirmed that, the next plan was precisely what you've suggested, that is to test the generality of this effect across different nuclear backgrounds, but in particular across nuclear backgrounds that have evolved in different parts of the Australian climate, so low latitude versus high latitude. And indeed, PhD student in my group who is a master's student with Prasad, Ekta, is doing just that for one of her PhD chapters, which we're currently writing and hopefully will be published in the next year. So that was indeed our follow-up and a great question.
Nice. Again from Amitabh, could the mitochondrial haplotype effects on male reproduction-related genes be an example of a reverse mate-harming kind of sexual conflict?
Prasad, can you read that question to me again? Sorry, I missed some parts of it.
Yeah. So could the mitochondrial haplotype effects on male reproduction-related genes be an example of a reverse mate-harming kind of sexual conflict?
Yeah, I think I understand the point here, and I guess some of the key points is the nuclear genes that are enriched with male, whose expression is enriched in the male reproductive traits such as the ejaculatory duct and the testes, they're only expressed in males, and they're seemingly interfered with by mtDNA. And it's possible those effects are clearly maladaptive. It's not clear whether they're just evolving under this quirk of maternal inheritance or whether they're at the heart of a sexual conflict. Because they don't really affect female gene expression so much, we had assumed that it was simply male maladaptive. But then when we started to assay reproductive traits, there was clear sexual antagonism. So the haplotypes associated with the highest female performance are associated with the lowest male performance. So I think there is some overt sexual antagonism, sexual conflict going on there. I'm not quite sure what you mean by reverse sexual conflict, but I would like to follow up in email or something.
I think most of the times one thinks of mate harm in terms of males decreasing female fitness, but this is sort of reverse. In that sense, it's a reverse mate harm.
I think in that sense that it's clearly reversed, although the females are unlikely to be wittingly trying to harm the male, but that also goes in normal sexual conflict as well. Typically, the female harm is a byproduct, a pleiotrophic consequence of intra-sexual selection in males. So in that sense, we can see it as reverse sexual conflict.
Excellent, sir. Thank you very much. Now, a few
so
very, very basic doubts. Very, very basic doubt. You have found claims for the haplotypes in mitochondria. Whether the nuclear genome is same in all these claims?
Can I ask, just for a clarification of question, Ranganath, are you talking about the claims or the clade?
You have found claims for haplotypes of mitochondria.
Yeah.
Right? Then, in the same cell, what is the status of the nuclear genome? Is it same in all the individuals of the cline, or the variations in the nuclear genome will have an impact on the cline in the haplotypes of mitochondria?
Yeah, it's a good question. So the nuclear genotypes cline as well. And while when it comes to neutral markers like microsatellite DNA markers, there's no cline, there's no variation across the cline indicating complete gene flow. When you look at functional loci, such as heat shock proteins encoded by the nuclear genome, they do strongly cline. So along that cline, we have many different traits clining. We have many different nuclear alleles clining. And we also now know that there's a mitochondrial cline that also maps to phenotype. When we wanted to show our validation that the mitochondrial haplotype is actually involved in the phenotypic clines we saw, then we put the mitochondrial haplotypes only along a standard nuclear background that lacked genetic variation, and we were able to link that at least some of that phenotypic variation along the cline does map to the mitochondrial haplotype in the predicted direction of the mitochondrial climatic adaptation hypothesis. So we had good evidence there. We have since conducted experimental evolution experiments where we evolved populations of fruit fly under cooler or warmer temperatures, and we allowed these two different haplogroups, the A1 and the B1 haplogroup, to compete under those two different thermal regimes. And indeed, it's a bit complicated. When there's no Wolbachia, which is an endosymbiotic parasite that is also maternally transmitted and cytoplasmically inherited in fruit flies, when there's no Wolbachia, indeed the mitochondrial haplotypes respond to thermal selection as predicted. The B1 haplotype outperforms A1 under colder conditions. A1 outperforms B1 under warmer conditions. When there is Wolbachia in the population, we don't get a clear-cut result as well. So things are more complicated once you have a more complex system. Our experiments try to simplify the system as much as possible, but it's a complicated world.
Okay. Thank you. The other question is, mitochondrial genome on expression of nuclearoid expression patterns. There are a couple of statements, effect of mitochondrial haplotype on the nuclear genome expression, right?
Yes.
What would be the reverse?
The reverse?
It says the impact of the nuclear genome on the expression of the mitochondrial genome.
Yeah, that's a wonderful question. We haven't looked at it yet when it comes to gene expression. We know that mitonuclear interactions are very important in changing phenotype, in moderating the phenotype. I do believe that the expression of key mtDNA protein coding genes will change across as we switch nuclear backgrounds as well. We just don't have that data at this stage.
My basic dilemma is, throughout the presentation, the mitochondrial haplotype is extremely important. But how far it is justifiable to ignore the contributions from the nuclear genome?
Yes, my feeling is that when it comes to complex phenotypes, that the mitochondrial contribution to that phenotype is going to be generally epistatic. That means that the interaction between the mitochondrial and the nuclear genotype will be one of the major contributing genetic determinants of phenotype. And many of our experiments, now that we've done this proof of concept, are testing just that to see whether, for instance, the male biases that we see in mitochondrial haplotypic effects on all of those traits that I showed, are they confirmed when we go across different nuclear backgrounds? And how important is the interaction of the mitochondrial haplotype and nuclear genotype? How important is that interaction in shaping the phenotype relative to the additive mito effects or the additive nuclear effects? Yep. We have some published data for that already. Yep.
Okay. Prasad, I have a last question.
Sure.
How about de novo mutations in the male itself? It did not come also from the female. No? After having got from the female, there could be de novo mutations in the mitochondria of males itself. Do you have any such report?
Okay, so I'll just ask for clarification again. You're talking about mutations in the mtDNA of males?
Yeah.
That are then somehow inherited.
Yeah.
So we generally, as we know, the paradigm is that in most animals, mitochondrial transmission is strictly maternal. However, there have been reported cases of leakage from the father. So reported cases of low incidence of paternal transmission of mtDNA, which would mean that if mutations were to arise in the mtDNA of a male, in theory it's possible for that male to pass it on to his offspring. But that would be very rare.
No, the question is different. The question is, the mutations that happen in male need not be passed on to the next generation. Agreed. But the mutations that happen in the male mitochondria, it can have impact on that individual itself, right?
Yes. So the generation, so basically, are you talking about interactive effects of on other individuals in which the mutation is not expressed? So for instance, it's possible, and I've got to apologize that I don't fully grasp the question at the moment. There are some technical issues at my end and it keeps cutting out a bit. But one thing we have been looking at is whether there might be capacity for selection to shape the mtDNA through males. Yeah, because, for instance, if a mutation was to arise in the mitochondrial DNA sequence that resulted in males that were superior when it came to helping or assisting their siblings, their female siblings or their parents, then you have indirect selection on the mtDNA, or vice versa, mutations that knock out male function then result in less resource competition for female siblings. And so you can have selection on mtDNA, on sex-specific mitochondrial effects that happen even in the absence of paternal transmission of mtDNA. I do feel that I haven't answered your question very well. I apologize for that.
Okay, thank you, sir. Thank you very much. Prasad, I'm through.
Yeah. One more question from Chantal. How do the environments of the other areas compare to Brownsville, which tends to be hot, humid, and relatively consistent throughout the year?
Yes, so we have haplotypes collected from all over the world, including equatorial areas, which are hot, consistently hot, with two seasons, humid and less humid, and then we have haplotypes from temperate areas as well. So Brownsville is not our only population that comes from a warm, a very hot, consistently hot haplotype, population. We feel that Brownsville mutation is probably not the major mitochondrial haplotype segregating in Brownsville. It was probably just a serendipitous collection, and one that we've retained because of its remarkable effects on phenotype. No one's been able to go out and recapture that from the field. That haplotype only exists in our laboratory, basically. But we know that it descends from Brownsville, originally collected from, I think, David Rand's lab, who's at Brown University in Rhode Island.
Okay. One final question, probably. Do we have any idea of the extent of mitochondrial genetic variation in laboratory populations? Because they are typically in the lab for a very long time. And if we do, how important are mitonuclear coevolution in the response that you see in laboratory selection experiments? Any idea at all about these things?
Yeah, we want to test this idea. So typically, I think typically when you go into any population of Drosophila, which is the species that we work with, you get low levels of variation. And that's reflected in the laboratory populations we collect from the field as well, because usually we start our mass-bred populations of flies from single locations, rather than pooling individuals from multiple locations across the globe. But still we see, even in the first example I showed you was a population which Larry Harshman collected called LHM. And we found segregating mitochondrial variation in that population some decade to 2 decades after the population was started in the laboratory. And we only sequenced 15% of the genome because back when we did that, it was quite labour and cost expensive to sequence mitochondrial genome. So that we were just trying to make sure that there was variation there, and we found at least 2 variants. We've had a look at other populations, and we have some low levels of mitochondrial variation, usually 2 to 3 haplotypes segregating. And as you can see, the sorts of variation we get in the Australian cline when we set up mass-bred populations from collection points on the Australian cline, typically we maintain both haplotypes over the long term in those mass-bred populations, which suggests that potentially there's some negative frequency dependent selection shaping those ultimately, and indeed some research out of Göran Almqvist's lab in Uppsala has showed in an experimental evolutionary context in a species of Drosophila and also in the Callicebrucus beetles that there is negative frequency dependent selection on mitochondrial haplotypes. So there is variation, but it's much less variation than you get obviously in the nuclear genome. We're talking maybe 2 or 3 segregating haplotypes typically.
Okay. I think we have answered all the questions that were there. So thank you, Damian, for a fantastic talk. This was really nice. And I thank all the participants today. Thank you so much, and hope to see you again later in the day for the next talk. Thank you, Damian. Thank you.
Thank you, Prasad, and thank you so much, everybody, for tuning in and for asking such informative and insightful questions that really made me think. And it's been a real pleasure. Thank you very much.